Hello! The purpose of this video is to show you how to properly dispose of all the different wastes that you'll generate over the course of your experiments in the lab. Indeed, there are several different types of waste that you may generate in the lab, and each one of them may need to be disposed of in its own unique way. For example, if you're unpacking a new shipment or something like that and you need to throw away paper or plastic wrapping that hasn't come in contact with any chemicals or cells, that can go in a normal lab trash can. However, if you generate any liquid chemical waste, that obviously cannot go in the regular trash can or down the drain. Instead, it must be collected in a glass collection vessel and properly labeled. As another example, if you generate any waste that's come into contact with cells, proteins, or DNA, that'll have to be collected in one of our red biohazard boxes. In addition to these examples, there are several other types of waste that you'll generate, each of which we'll go over in this video. And please remember, many of the wastes that you'll generate could be harmful to yourself or to the environment. So please dispose of chemicals properly and ask your supervisor if you are ever unsure of how to dispose of something. The simplest type of waste that you'll deal with is cardboard or plastic wrapping that's used for shipping. These things can be put in the regular waste disposal. Large pieces of cardboard waste can be put out into the hallway, where they will later be collected for recycling. However, if any of these materials, paper, plastic, or even cardboard, come into contact with chemicals or cells, then they must be treated as chemical or biohazardous waste, which we will cover later in this video. The most common type of waste you'll generate is liquid chemical waste. This could be an acid or base solution, maybe even a dissolved metal. In any case, liquid chemical waste should be collected in a glass vessel with a lid. These chemical waste containers are usually kept inside or very near the chemical fume hood. Whenever you add a chemical to the bottle, make sure you update the label by writing the full scientific name of the chemical. Also, before adding a chemical to the bottle, check the label to make sure there won't be any harmful reactions between the chemical you're adding and chemicals already present inside the bottle. For example, you should always try not to mix large amounts of acids, bases, and halogens together. If you're ever in doubt on whether or not it would be safe to add a chemical to the waste bottle, contact your supervisor. From time to time, you may also accidentally break a glass container in the lab. If this happens, do not attempt to clean it up with your bare hands or even gloved hands. Instead, go get a broom and a dustpan from the janitorial closet. Take your time when sweeping and make sure you get as many of the little glass shards as possible. Once you've collected all of the broken glass, dispose of it in one of the cardboard boxes labeled broken glass. Remember, any piece of glass with even the slightest crack in it is very unstable, especially if you're going to expose it to different temperatures. Therefore, if you have a beaker that has even the slightest crack, it should be disposed of in the glass waste box immediately. Over the course of your experiments, you'll probably use pipette tips and syringe needles at some point. If you use a pipette tip, it must be disposed of in one of the sharps boxes shown here. Pipette tips cannot be disposed of in biohazard bags because they may poke through. If you use a syringe needle, make sure you put the plastic cap back onto it, then unscrew it from the syringe, and dispose of it in the sharps box. If you ever use a pipette tip to transfer a hazardous chemical, for instance a strong acid, make sure you neutralize that acid with a base or dilute it with water before disposing of the tip in the sharps box. If your pipette tips or needles are exposed to cells or any other biohazardous wastes, they must be sterilized before we can dispose of them. The best way to sterilize a sharps box is to autoclave it along with other biohazardous waste at 121 degrees C for 30 minutes. Once your sharps box has been sterilized in the autoclave, it can be disposed of with all the other biohazardous waste. Speaking of biohazardous waste, any lab that does biological research is required to have a biohazardous waste box. At all times, the biohazard waste box should be lined with a red biohazard waste bag. Most kinds of dry biohazardous waste can be disposed of in these red boxes. For example, you might use them to throw away pipettes, empty 15 or 50 ml tubes, paper towels or chem wipes, and even the latex or nitrile gloves you use on your hands. If you generate any biohazardous waste that contains a liquid, that liquid must first be removed before you can use the biohazard box. In this case, we're removing the cell culture media from a T75 cell culture flask before putting it in the red biohazard box. Also note that the Pasteur pipette I used was glass, so it has to be disposed of in the glass waste box. Unfortunately, our autoclaves aren't large enough to fit full-size biohazard bags. Therefore, you should always try to seal off the bags when they get about halfway full. If a bag gets more than halfway full, transfer the excess to a new bag. Once your bags are ready, carefully detach them from the brown cardboard box and evacuate as much of the air from the bag as possible. Do both of these steps very carefully and slowly, since it is possible to tear these bags. Once you've gotten as much of the air out of the bag as possible, twist off the excess plastic and secure it with a piece of tape. If you have two biohazard bags that are both halfway full, you can transport them in the same biohazard waste box. Make sure you use a cart to transport the biohazard waste box over to 111 Mendel, where the autoclaves are. Each autoclave can handle one biohazard waste bag at a time. Make sure you put the bag in one of the large plastic bins, just in case there is any liquid in it and it leaks out. Sterilize the bag by autoclaving it at 121 degrees C for at least 30 minutes. Remember, autoclaves use steam, which is hazardous, so do not open the autoclave until the cycle is fully complete. Once the cycle is complete, put the red bag back into the biohazard waste box and seal all sides of the box with packaging tape. Once the box has been properly sealed, take it down to the basement of Mendel to dispose of it. Ideally, biohazard waste boxes should be collected in room G04, but if the door is locked, you can leave the box outside of the door. If you ever work with any cells, you'll have to properly dispose of them as soon as you're done with your experiment. Please try to dispose of old cell cultures as soon as possible to prevent them from building up in the lab and providing breeding grounds for other types of contaminants. If you need to sterilize an animal cell culture, bring it into the BSC. There you can aspirate all of the liquid cell culture volume into the aspirator tank. Since the aspirator tank always contains 10% bleach by volume, it should instantly sterilize the old cells. When the aspirator tank fills up, you can take it out of the hood and dump it down the sink. However, remember to put 100 to 200 mL of bleach back into the aspirator tank to keep it sterile. And as we already mentioned, dispose of any glass Pasteur pipettes you use in the glass waste box and any dry cell culture materials in the red biohazard bag. Small bacterial cultures can also be sterilized with 10% bleach. Simply add your bacterial culture to a glass bottle, then add an equal amount of 10% bleach. Transfer the bleach cell culture mixture to the hood, just in case it gives off any harmful vapors. After the culture has been sitting in bleach for at least 24 hours, you can dump it down the drain. Please also remember to clean out the glass bottle you used as soon as you're done with it. Larger bacterial cultures should be sterilized in the autoclave. Make sure you put them in a secondary container just in case they spill out. As with any other liquid, autoclave these cultures at 121 C for at least 30 minutes. Once the culture has been autoclaved, you can safely pour it down the drain and rinse out the flask. Speaking of flasks, it is important to mention that bleach can damage polypropylene, which is the material that many of our flasks are made out of. Therefore, if you have a bacterial cell culture in a polypropylene flask, please do not sterilize it with bleach as it will damage the container. Instead, you should always autoclave polypropylene flasks. And while we're on the subject, please remember to never autoclave bleach since it creates toxic vapors. Last, but definitely not least, is ethidium bromide. Ethidium bromide is a dye molecule that's commonly used in biological experiments involving DNA. The ethidium bromide dye, also known as ETBr, strongly fluoresces when it binds to DNA molecules. While this is very handy in visualizing DNA molecules in, say, an agarose gel, it's also very hazardous since ethidium bromide can also bind the DNA in your cells and interfere with gene expression. Therefore, ETBR is classified as a carcinogen and should be treated with extreme caution at all times. Ethidium bromide waste should also be collected separately from other chemical waste since it needs to be treated prior to disposal. For example, if you have any agarose containing ethidium bromide, make sure to contain them in a separate box. Any liquid ethidium bromide waste should also be collected in its own tube and labeled properly. If either the dry or liquid ethidium bromide wastes fill up, let your supervisor know immediately. So, let's review how you should dispose of all these types of waste. If you have uncontaminated normal waste, such as plastic wrap or paper, that can go in the normal lab trash can. If you have liquid chemical waste, that has to be collected in a glass bottle, which is normally inside or near the fume hood. If you accidentally break some glass, collect it with a broom and dustpan and dispose of it in the glass box. Other sharps, including pipette tips and needles, should be collected in a hard plastic sharps box. Remember, if these tips and needles have contacted any cells, they should be autoclaved and disposed of with the other biohazardous waste. Speaking of which, all dry biohazardous waste should be collected in the red biohazard box and autoclaved. All liquid biohazardous waste, including used bacterial and animal cell cultures, should either be autoclaved or sterilized with an equal volume of 10% bleach, and then poured down the drain. Finally, special types of waste, such as ethidium bromide, should be collected in their own waste containers that are properly labeled and disposed of separately from other chemical wastes. Remember, if you have any doubts or uncertainties on how to dispose of a waste that you generate, contact your supervisor to find out what you should do. Otherwise, you might harm yourself or the environment. So that's about all we have for this video. I hope we didn't waste your time, and I wish you the best of luck in your research.